This is another enzyme with a different active site to the previous one. And these here are substrate molecules. So enzymes can break molecules down into smaller molecules, or enzymes can build larger molecules from smaller molecules. So what this enzyme will do is build a larger molecule from these substrate molecules. So the substrates will bind the active site, and the enzyme will build larger molecules from the smaller molecules. So as you can see here, a larger product is made. So this is the enzyme. This is the active site of the enzyme. These are the substrate molecules. So these are the substrate molecules that bind the active site of the enzyme, which then form the enzyme substrate complex. And this here is the product, what is made. And this is still the substrate because it hasn't been changed into a different product. Enzymes are biological catalysts. They speed up the rate of reactions in your body. Proteins are formed of amino acids bonded together. And it is the enzyme protease that binds the substrate protein. It is the enzyme protease that breaks down proteins into amino acids. This here is a molecule of starch, the polymer starch. Starch is a carbohydrate, and it's broken down into glucose molecules, which is used by the body for respiration. It is the enzyme amylase that binds the substrate starch. Starch is the substrate and amylase is the enzyme. Amylase breaks down starch into glucose. Fats, aka lipids, also need to be broken down by enzymes in the body. So what I'm showing you here is an enzyme binding a lipid, aka a fat. The enzyme that binds the lipid is called lipase. Lipase binds the lipid. Lipase, lipid. And the lipase breaks down the lipids into fatty acids and glycerol. So glycerol and fatty acids. That is, so if you increase the pH for enzyme A above or below pH 2, the enzyme's active site will denature. The further away from pH 2 you change the pH, the more the enzyme's active site will denature. And the more the enzyme's active site denatures, the more difficult the substrate will find it to bind the active site, so therefore less product will be formed. So at pH 3 in this example, the enzyme's active site has fully denatured. This means the substrate cannot bind the enzyme's active site at all, which means the rate of reaction will be 0. No product will be produced because the enzyme's active site has fully denatured. This point here is where the enzyme is working at its fastest rate. At this temperature, this is the optimum temperature where the rate of reaction is at its highest. So the rate of reaction is at its highest for this enzyme at this temperature, which is 37.5 degrees Celsius. The optimum temperature, which is 37.5 degrees Celsius, is the temperature where the rate of reaction is at its highest. And in this case, it's 37.5 degrees Celsius. So if we increase the temperature from 37.5 degrees Celsius, which is the optimum temperature, and we increase it above the optimum temperature, in this case to 50 degrees Celsius, the rate of reaction decreases. So the rate of reaction will decrease. So at its optimum temperature, the enzyme is breaking down or making products at its maximum rate. So as you increase the temperature above its optimum temperature, the active site of the enzyme denatures. It denatures, it changes shape. Therefore, less substrate can bind the active site of the enzyme. And at this point, even less substrate can bind the active site of the enzyme. So if less substrate binds the denatured enzyme's active site, that means less product is produced, which means the rate of reaction decreases. So if you decrease the temperature below the optimum temperature, then there'll be a decrease in the kinetic energy of the enzyme and the substrate. so they're going to move slower. So as you can see here, there is going to be a decrease in the kinetic energy store of the enzyme and the substrate. So at this point here, at the optimum temperature, this is where there are going to be more collisions between the enzyme and the substrate than at a lower temperature here, where there are going to be fewer collisions between the enzyme and the substrate because they are moving slower. There is lower kinetic energy in their kinetic energy store. So if they are moving more slowly, that means there's going to be a decrease in the collisions between the enzyme and the substrate. So if there are fewer collisions between the enzyme and the substrate, that means the rate of reaction decreases. It decreases. Less product will be made because there are less collisions between the substrate and the enzyme.